Hello everyone, my name is Qing and I'm one of the study directors with expertise in the transporter-related area. In the following slides, I'm going to use several case studies to show the difference of the compound if different systems are chosen. With the introduction of new draft guidance by US FDA, people have realized the importance of in vitro systems to differentiate the drug-drug interaction. Therefore, people have heavily relied on the interpretation of the in vitro results of the interested compounds. There are many systems available through the different CROs and pharmaceutical companies. Are those systems equal? Will the same compound have the same results if use different systems? These questions are of critical importance for the sponsor of the new drug to know. In absorption systems, there are two parallel cell-based assays available. The first set is Caco2 and Caco2 derivatized cells. These derivatized cells are patented with a reduced expression level of some selected efflux transporters, including P-glycoprotein (PGP), breast cancer resistance protein (BCRP), and the multiple drug resistance protein (MRP2). These cell lines, in origin, were from human colon carcinoma cell lines. These cell lines are also from human, with a number of efflux and uptake system present in Caco2 and its knockdown derivative cells. The second set of system is MDCK transfected with human PGP gene and also its control, MDCK cells. These cells with a highly expressed level of PGP, but these cells are from dog, it's not human. The decision tree about whether the compound is a PGP substrate or not is really straightforward. If the compound have efflux ratio in either systems greater than 2, then yes, the compound may be a PGP substrate. The next step is to challenge the compound with a specific PGP inhibitor. If the inhibition was greater than 50%, the compound may be a PGP substrate. It looks very straightforward. However, in different case studies I'm going to show later, you will see the complexity of the compound as well as in the systems. In this slide, the blue bar is Caco2 over the CPTB1. CPTB1 is the cell line with the reduced BCRP expression. We can see between two different systems, there are agreements. For example, like SN38 and quinidine. However, there are also controversial. For example, rosuvastatin didn't show any efflux in BCRP transfected MDCK cells, but shows efflux in Caco2 cells. And prazosin, which saw efflux in the Caco2 over CPTB1 cells, but much higher efflux in the BCRP MDCK systems. There are also some other compound like digoxin showed a borderline results in two different systems. Again, this slide shows the the compound in different systems. Fexofenadine, this is clinically proven to be a PGP substrate, and it also showed efflux in Caco2 versus CPT-B1 cells, but it didn't show any efflux in MDR1 MDCK cells. Loperamide and labetalol are PGP substrates, but the generated efflux response are quite different between these two cells. So why are the results of the same compound so different in different systems? This basic cartoon shows a oversimplified pathway that one compound can take to permeate through the cells. JC stands for carrier-mediated drug transport. JM stands for the passive diffusion. JP and JT are relatively negligible in drug transport through membranes. So when the compound happens to be the substrate of the transporter, the carrier-mediated transporter component will dominate all the other components. The compound will show a transporter-specific property. Well, if the compound has a really high passive diffusion, then this component will be standing out, and all the other components will become less relevant. This one is also from one of our sponsors. The compound shows efflux ratio about 3. Clearly, this compound appears to be a PGP substrate. Theoretically, a number of complete studies would be waiting for the sponsor to carry on to get a complete knowledge of this compound. However, the apical to basolateral apparent permeability was very high. It's about 10 times 10 to minus centimeter per second. We have developed our unique high permeability internal standard, HPIS. And this HPIS has the apical to basolateral apparent permeability approximately about 4 times 10 to minus 6 centimeters per second. Very obviously, the sponsor's compound beats this HPIS. Well, the HPIS, if higher than 4, the absorption potential in human is closely to 100%. Very likely, the sponsor's compound is also going to have high absorption potential. Therefore, the efflux property of this compound will be overlooked by the high permeability potential of the compound. Both FDA and EMA realized and acknowledged this high absorption potential, which can mask the effect of the efflux on the absorption potential of the compound. Still, I'm using the rosuvastatin as an example. The compound shows a high efflux ratio in Caco2 cells. Another cell line, LRS513, was used. This LRS513 cell line was also originated from human carcinoma. The difference between the Caco2 and LRS513 is on the basolateral expression of OST. When Caco2 cells were challenged with OST inhibitor, taurocholic acid, TC, the efflux ratio of rosuvastatin decreased significantly. Also, when Caco2 cells were treated with OST antibody, the efflux ratio of rosuvastatin also decreased. In RS513 cells, since the OST expression level was relatively low with the treatment of the taurocholic acid, the efflux ratio didn't change compared to without the treatment of the taurocholic acid. Also in MDR1-MDCK cells, there is no expression level of OST ever found. As such results, the rosuvastatin didn't show efflux in MDR1-MDCK cells. These slides indicated the presence of basolateral uptake transporter may be necessary for certain compounds to show efflux property. Cladribine is also clinically used a compound. Initially, in the MD1-MDCK cell systems, cladribine appears to be a PGP substrate, but in the Caco2 over CPT-P1 cells, the efflux ratio of cladribine is close to 1. CPT-P1 cells are cells with a reduced expression level of human PGP. In the clinical environment, cladribine didn't show any PGP-related property. However, it does show interact with the BCRP substrate. The cladribine in vitro systems shows to be also a non-PGP substrate in Caco2 systems. In this case, Caco2 over CPT-P1 displays a better in vitro in vivo correlation. This is another case study with a sponsored test compound. It again shows high efflux ratios in MDR1-MDCK over MDCK cell systems. It didn't show efflux ratio greater than 2 in either Caco2 or CPT-P1 cells. Therefore, the compound is not classified as a PGP substrate if using Caco2 cells. With further information disclosed by the sponsor, this compound tends to be target tumor cells, while the tumor cells have a high expression of a PGP. So with the information of the compound in MDR1-MDCK systems, the sponsor may be able to use structure-activity relation software or different models to further optimize the structure of the compound, trying to reduce or eliminate the PGP properties of the compound with a high availability to the tumor cells. With the different compounds as shown in this presentation, each system has disadvantages and advantages. Both of them tend to give false classification. However, the Caco2 cells offer a broader scope of the compound. For example, if the apical to basolateral permeability of the compound in Caco2 systems are high, this compound maybe could be used as a waiver in the BCS classification. And also, with the multiple systems present in the one cell line, the Caco2 system tend to give a better in vitro in vivo correlation. On the other hand, MDR-MDCK cells with a highly expressed single transporter, PGP, sometimes these systems would exaggerate the efflux property of the test compound. Because this system has only one enhanced expression of PGP, sometimes this system tend to give a not accurate in vitro in vivo correlation. Thank you very much for your time. If you would like to get more information on drug transporters, please visit our website at www.absorption.com/transporters. Also, if you are interested in getting a poster from this presentation, please visit www.absorption.com/poster. Again, thank you very much for your time.